When the body recognizes that red cells are being lost too rapidly, the bone marrow ramps up production to compensate. This response is called increased erythropoiesis and produces several identifiable findings. In the marrow itself, there is normoblastic erythroid hyperplasia, meaning the marrow is packed with precursor red blood cells working overtime to replace what is being lost. In the peripheral blood, this marrow effort shows up as reticulocytosis, an elevated count of newly released immature red cells called reticulocytes. These young cells still contain residual RNA from their time in the marrow, which makes them identifiable on special staining. Because reticulocytes are physically larger than mature red cells, a high reticulocyte count can push the mean corpuscular volume, MCV, upward slightly, up to approximately 105 femtoliters. This is important to recognize because it can make a microcytic anemia appear falsely normocytic if reticulocytes are abundant. A related blood smear finding is polychromatophilia, where these larger, slightly bluish-staining, immature red cells are seen alongside normal ones. This is essentially the morphologic equivalent of a high reticulocyte count and reflects the same surge in compensatory output. Regarding classification terminology, when the underlying problem lies within the red cell itself, such as a membrane defect or enzyme deficiency, this is called intracorpuscular hemolytic anemia, and these defects are most often genetically inherited. When the problem originates outside the red cell, such as circulating antibodies or an enlarged spleen trapping and destroying normal cells, it is called extracorpuscular hemolytic anemia, and these causes are most often acquired. In summary, erythropoiesis is the highly regulated process by which hematopoietic stem cells in the bone marrow differentiate into mature red blood cells, RBCs, through a series of progenitor and precursor stages, primarily under the control of the hormone erythropoietin, EPO, which is produced by the kidneys in response to tissue hypoxia. The process requires adequate supplies of nutrients like iron, is influenced by local bone marrow microenvironmental factors, and is regulated at various stages by transcription factors, notably GATA1, cytokines, and cell-cell interactions, with feedback mechanisms between erythroid cells and iron metabolism to regulate erythropoiesis. Erythropoietin EPO is the central hormonal regulator, promoting proliferation and differentiation of erythroid progenitors. Its release is triggered by hypoxia and suppressed as oxygen delivery improves. Stages of erythropoiesis. Development progresses from burst-forming unit erythroid BFUE to colony-forming unit erythroid CFUE to proerythroblasts and ultimately reticulocytes, with EPO acting mainly on CFUE and later stages.